I am Cadet Jennifer Cabanas, and my colleagues, Cadet Gonzalo Cacho and Cadet Taylor England, have been conducting research using time-lapse imagery of malignant melanoma cells with the help of our advisor, MAJ Stephen Hummel, at the United States Military Academy at West Point. Our project is called Identification of Chemical and Biological Agents Through Cell-Based Screening. In the next few minutes, my colleagues and I will explain the purpose of our research, the background, general methodology, recent results, and future direction. The purpose of our project is to find a novel method of identifying chemical or biological agents exposed to soldiers in the field. Our hope is to speed up the process of identifying morphological changes in the cells.
Currently, methods used to detect chemical and biological weapons are not adequate for a rapidly shifting battlefield. For example, most tests to detect biological agents can take close to 96 hours before they are able to identify the target, which could put our soldiers at risk. So basically, your project is trying to figure out a faster way of doing this. And how it works, we're basically putting cells under microscopy, and then we are trying to see the motility, proliferation, and just general cellular morphology, and analyze those different traits and see if we can set up a standard to later on be able to detect the agent.
In order to conduct this research, we do a couple different things. The first is tissue culture. We take 8375 cells that are maintained and monitored in order to keep passage number low and to ensure cells are readily available for image microscopy, where we take 8375 cells and they're seated at low density in 96 well plates. Each plate will have untreated controls as well as dose-dependent wells. The plates are then imaged using Nikon Eclipse TI inverted confocal microscope with real-time capture and environmental chamber every 10 minutes for 7 days. The third thing we do is image analysis. We acquire images and analyze them via MATLAB such that each cell in each frame is identified, morphological features measured, motility measured from frame to frame, and death proliferation rates are taken. The last thing we do are bioinformatics linking, which analyzed image data based on agent and dose exposure with a compared and linked to any pre-existing protein and gene expression data sets in order to understand the process from time of exposure to cell death.
Furthermore, to increase our analysis capabilities, we're trying to see the 3D space of the cells in the plate. And this we're doing it with a T-distributed stochastics neighbor embedding, or TSNE. So how this works, we conduct a ANOVA of different morphological features of the cells at each time point and then normalize the values. And this allows us to look at things that don't normally appear under microscopy. Our results have demonstrated that morphological changes can be graphed, and we can identify differences in cell morphology when a drug is added. And statistical differences are obvious, and with further research we can probably detect the drugs. Following the development of biosensors capable of immediate bioweapon detection based on characteristic cell signatures, the eventual goal of the project is to aid in the creation of a biosensor in the form of a human on a chip. Like the canary that once was used to alert miners of deadly gas in coal mines, differentiated human cells on a chip could be rapidly analyzed by a biodigital canary biosensor to determine the type, duration, and concentration of bioweapon exposure in order to initiate timely and appropriate treatment.